Good to go.
You ready to go? All right, good morning, everybody. Pleased to introduce to you Dr. Jason Pitts from Baylor University. And he comes from the Midwest. His home is Indiana. He got his bachelor's in biology and genetics at Ball State University. Anybody know where Ball State is? Well, you're going to tell me?
I don't know. I know it's in Indiana.
Oh, okay. All right. It's in Muncie. All right. So, and then he got a master's degree at Indiana in Bloomington in plant sciences, and then a PhD at Vanderbilt in Nashville, Tennessee, where he got also his degree in biology as well as molecular biology. He was there for nearly 20 years as a research olfaction sensory expert, and he's going to give us, I think, a really good talk about how insects actually interact with the environment. But before we do that, why don't we say a short prayer, and we'll let Dr. Pitts get going. All right. Lord God, thank you so much for the opportunity to have Dr. Pitts here with us. I just really pray that you just help him teach us the things that are of beauty, mystery, and of your creation. Ask this in your name. Amen. All right.
Well, thank you. I really appreciate the opportunity to be here today with you. This seminar can be highly interactive, so feel free to ask questions along the way. I've tried to tailor this talk in such a way that for those of you who may be engineering students or chemists or otherwise, that the biology isn't too overwhelming. So again, feel free if there's something that you don't understand, just ask away. So the title of my talk, as you can see here, is Chemical Detection Systems for Insects. I'll primarily focus my talk today on mosquitoes. So my lab I would call is a vector biology lab. So we are interested in mosquitoes primarily because they are such important transmitters of pathogens that cause human disease. I'm going to start with acknowledgments, if you'll indulge me. I'd like to make sure that I credit the people who are doing the work in my laboratory and our collaborators with their contributions to the project that I'll describe. So here is a picture of my lab in 2018. I arrived at Baylor in 2017. So this was the first group of students that were in my lab. There are many undergraduates in here as well as graduate students. This is the lab as of last year in 2022, maybe a bit smaller. Some of our undergraduates have graduated and moved on to other things. But we are funded by the National Institutes of Health, the NIAID specifically. We have two projects funded by the NIH, one from the American Mosquito Control Association. I'd like to highlight just a few of our collaborating colleagues. At the top here is Dr. Jonathan Bobat, who's at the Hebrew University of Jerusalem. He and I have been friends and close colleagues for many, many years and have published lots of work together. Doctors Jeff Riffle and Omar Akbari are at the University of Washington and UC San Diego respectively, and we have an ongoing project that I'll describe to you today to try to understand better how mosquitoes detect floral odors for the purpose of nectar feeding. Did you know that mosquitoes feed on flowers, feed on the nectar from flowers? You think of them as pollinators. Many people don't. They just think of the nuisance of mosquitoes and how important they are as disease vectors, and that's true. But we want to understand all aspects of the chemical ecology of insects broadly and mosquitoes specifically, so that's what this slide is meant to depict for you. Insects, maybe surprisingly, lead kind of complex lives. They do lots of things in their time. Now, that could vary, of course, across insects, broadly speaking, but mosquitoes in specific, and female mosquitoes particularly, engage in lots of complex behaviors. And they do so largely through detecting chemicals in their environment. So what my lab is really focused on is trying to understand at the molecular level how it is that mosquitoes can detect and respond behaviorally to compounds in their local environment. And they do that because they have to exploit natural resources in order to survive and reproduce. That's fitness in a nutshell, right? And so mosquitoes will seek out flowers for nectar feeding. Both males and females will do that. Females will seek out hosts, like you and me, for the purpose of blood feeding. Females need the protein and lipids in blood in order to produce eggs for reproduction. Females will also find adequate water sources for oviposition, that's egg laying. And even at the larval stage, so there's an aquatic stage, the life cycle, larvae are chemotactic. They have to find food in their environment that they can feed on, and they store nutrients in order to metamorphize as pupa and eclose as adults, and then utilize those resources that they've stored in specialized organs early in their adult life stage. And then finally, across insects, broadly speaking, there are chemical detection systems that are required for mating. Okay, so how do conspecifics find one another for the purpose of reproduction? Now there's scant evidence in mosquitoes, I'll admit to you, that there are chemicals that are given off pheromones, so-called pheromones, for the purpose of one member of the species finding another, but we think there's good reason to believe that those kinds of chemical interactions also exist in mosquitoes. And they can be important avenues for us to, and I'll explain this maybe a little bit at the end of the talk, for trying to interfere with the biology of mosquitoes and other pest insects. So maybe you're very well aware, I hope that you are, that vector-borne diseases are still a major burden to human health, human and animal health across the world. So this is just a schematic kind of diagram showing intensities of dengue transmission or dengue risk around the world on a country-by-country basis. You could see from a map like this that it's countries in the tropics and in the subtropics that still have major endemic transmission of dengue virus. We would call it dengue and arbovirus because it's transmitted by arthropods, including Aedes aegypti, which is one of the major vectors for dengue around the world. So the WHO says that about half of the world's population is at risk for dengue on an annual basis. And there are literally hundreds of millions of cases. Now often dengue is not a fatal disease to people, but it can be very debilitating, and there are different what we call serotypes of the virus. And if a person acquires a second infection of dengue of a second serotype, that often leads to hemorrhagic fever and is very fatal for people. So there's reason for us to study mosquitoes, not just because of dengue, because of malaria, from West Nile virus, lymphatic filariasis. There are a large number of diseases and pathogens that cause disease, I should say, that mosquitoes transmit around the world. So we're interested in understanding the molecular details of chemical detection because they're so critical for those behaviors that lead to disease transmission. So the patterns of diseases, it should be no surprise then, correlate with the distributions of vectors like Aedes aegypti. Here's a female Aedes aegypti. Maybe you've seen them. They're right here in central Texas. They are invasive in many parts of the world. This map is depicting the range of Aedes aegypti that's presumed to be viable for them. This doesn't mean that they're found in all these places. We can see that Aedes aegypti is quite viable right here in Texas. In fact, I have them in my backyard, and so we collect mosquitoes sometimes to bring into the laboratory so that we can study wild populations of them. Sounds dangerous a little bit, doesn't it? It's not, because they're not infected here in Texas, generally speaking, and at the larval stage they don't tend to be infected. It's only at the adult stage when females pick up infected blood that they become infected. So it's an invasive species. Does anybody know anything about the history of the invasiveness of Aedes aegypti by chance? How is it that it spread from its original native range to other parts of the world? There's some interesting literature out there, you might look into this, but the slave trade, actually. So 400 years of slave trade brought Aedes aegypti, it's thought, to the Americas. Yeah, and there's some really interesting history about yellow fever. Aedes aegypti, for a long time, is commonly known as the yellow fever mosquito. And so in the late 18th century in the United States, in cities like Philadelphia, Memphis, Tennessee, New Orleans, Boston, there were major outbreaks of yellow fever. In just a short period of time, 10% of the population of Memphis, Tennessee died from yellow fever. Talk about a pandemic. Can you even imagine that? Like 10% of a major city, the population died from yellow fever. At that point in time, no one knew that mosquitoes, let alone Aedes aegypti, were transmitting the virus that caused yellow fever. But of course, there were lots of measures that were taken to try to prevent its spread, including just stationing horsemen on the outsides of cities to prevent people from getting in or getting out because they didn't know where this was coming from. So it's kind of a remarkable history. And it's largely through human activity that Aedes aegypti has been spread and other vector mosquitoes around the world. Just a brief sort of description of modes of pathogen transmission that I think will help you understand the talk a little bit better. So there are a few things that are required for this kind of vector-borne disease transmission cycle. The first is a population, in this case of people, that are susceptible to a given pathogen. So not all pathogens, for example, of animals are infective to humans and vice versa. There are some that can move back and forth, of course. So a permissive host means one where the virus or whatever the pathogenic agent is can't replicate within that host. Okay, and then there's the pathogen itself, so that has to be infective to the host, and the vector must be competent for that pathogen. That is to say that if a female takes up a blood meal in which that pathogen is found, then the pathogen should be able to replicate to reasonably high numbers and also migrate to the salivary glands. It's in the act of biting where a female not only acquires the pathogen, but after she bites again at some period of time later, this is called the extrinsic incubation period, that she, through her salivary glands, transmits that virus or pathogenic agent to her next unwitting victim, whoever that may be. So there's a timing here that's important, and I hope I get to that to tell you why that timing is important to some of the aspects of disease vector control that we're trying to develop in my lab. Okay, so back to this chemical ecology slide then. So we're interested in all aspects of chemical ecology. I'm going to focus on two main themes today. The first is, as I've already alluded to, the molecular mechanisms of chemical detection. How is it that mosquitoes or any insect, how they're able to detect very specific chemical compounds that are given off in the local environment? We want to understand that really at the most nuanced level, at the level of protein receptors that are in sensory neurons. I'll explain more about that in a moment. And then how can we use this information to our advantage? How can people use this information globally to our advantage to reduce disease transmission cycles? Largely that would be through better surveillance and population control. So I need to explain just a little bit about the molecular biology of chemical detection. Okay, so bear with me on this. I'll try to make this pretty straightforward and simple. But if you were to look at the head of a female mosquito or any insect, of course you know that insects have paired antennae. And if you were to zoom in on the antennae, what you'd see are little tiny sensory hairs. They're called cincilla. And then if we were to slice open a single cincillum and look inside, what we would find is a fluid-filled cavity that has what are called ORNs, or olfactory receptor neurons. There are usually multiple neurons in those cincilla, 2 to 5, and depending on the insect, there can be as many as 50 of those in one. Not in mosquitoes, just a few. And each of those, if we were to zoom in a little bit closer on the membrane that projects into this fluid-filled cavity, they express proteins. So these are encoded by genes in the genome. And each of these proteins, we think, is tuned, if you will, or sensitive to a particular chemical compound, or maybe even with some variation, a group of chemical compounds, and that's what's depicted here. So this would be the membrane surface. There are large families of these receptors encoded in the genomes of insects, and mosquitoes specifically. They have lots of interesting names, but I'm going to focus in on this group here, called the odorant receptors, or ORs for short. So if you hear me use that term OR, that means odorant receptor. And here's how they function. A ligand, or a chemical compound that's carried by air currents, enters through small pores in this cincillum and then makes its way to the surface of this receptor. Now, if this receptor has evolved to be sensitive to that compound, this is what's called a ligand-gated ion channel, and it will literally open a pore in the membrane and allow charged ions to flow into this cell. The repercussion of that is if there's a strong enough change in this part of the cell, electrical change, that will lead this cell to fire what's called an action potential. And that's propagated down the axon back into the central brain of the organism. And that is a mechanism by which at the sensory peripheral level, mosquitoes and other insects can become aware of the presence of a compound. That compound may be part of a blend of compounds that signals a resource that's available in a local environment. Does that all make sense? Are you with me so far? Good, okay. So we're interested in this. What are the genes? What are the proteins? And what kinds of compounds do they respond to? Now, I'm going to tell you a bias that I have right off the bat here today. And that is that I believe that these proteins are really very, very specialized to detect one compound with highest sensitivity, that is the lowest amount of that compound possible in order to generate this action potential that leads to a behavioral outcome. There are others in my field who believe that these are more generalist in their approach, that they would respond to a whole suite of different compounds. I think that just generates noise in the system. So what I'm going to tell you today, and just to admit up front to you, this bias is that I think they're very, very specific, and I think we're finding evidence to suggest that they are specific, not only to a particular chemical compound, but even to isomers of those particular compounds. And that's important, I think, for discrimination of potential sources in the natural environment to mosquitoes and other insects. This looks a little bit busy, but before I tell you how do we do this, I need to describe the experimental system that we use to test hypotheses around chemical detection. We use, to put it quite simply, unfertilized frog eggs. Yeah, does that surprise you? It's a heterologous cell system. Well, what this frog egg can do for us, it has several advantages. One, it's a giant cell, not as giant as this, but it's giant. About 1 millimeter in diameter, these little spherical cells. So they're easily observable with the naked eye, they're easy to manipulate experimentally, and importantly, we can inject these cells with any gene from a mosquito that we wish. And they're just like little factories. They will produce those proteins, and those proteins will become expressed at the surface of the membrane, and they will function as ligand-gated ion channels that open and close, and open in response to chemical compounds that we challenge them with. Then we can just take a couple of little a couple of electrodes here and measure changes in electrical potential across that membrane. And so we have a perfusion system, a liquid perfusion system that constantly flows over the oocyte, and pre-injected with a gene of interest to us, a channel, an photoreceptor, whatever that may be. Then we begin to add in, at specific time points that we know and we can control, chemical compounds that we think might activate a response. And this is a list of chemical compounds. It's what I call our odor library in the lab. We selected these compounds because they have known behavioral responses in insects, maybe not just mosquitoes, but broadly. They're fairly ubiquitous in nature, so from floral odors to water sources to human sweat, these are the kinds of compounds that we want to find. Okay, and so we're using this as a way to narrow down the list of potential candidates. Can you imagine how many chemical compounds are out here on your campus, right, that are possible for an insect to detect? Again, that represents noise in the system. Insects, and specifically mosquitoes, should be able to really focus in on one or a few compounds that really identify a natural resource that they have to have in order to survive and reproduce. So that's why we've designed our chemical library this way. Back to the experimental setup then, so if we happen to challenge this receptor that's at the membrane surface of this oocyte with a chemical compound that activates it, it will open, positive ions will flow in, and on our little computer we can see an inward current. That's what that little deflection represents. It's a change in conductance across the membrane. It's actually super cool. And when this works, like when you see it happen, it's just fascinating. I've been doing this for 20 years and it still fascinates me every single time because it is the point of discovery. It's the point when we've learned something totally new about a receptor that maybe we had a particular hypothesis about, it's sensitive to some floral odor, and we find out if that's the case, can we support that hypothesis? Or we find out something totally new because we have this approach where we, in blends actually, we challenge this receptor with 200 compounds and we can just ask, okay, this one maybe not, but something else may show up too. And that's really, really fascinating, so love it. Okay, so chemical ecology of mosquitoes. This is some work that was published by our collaborators, Jeff Riffle and Omar Akbari. And I'm using this just to illustrate a point here with a knowledge gap that we have in terms of chemical detection biology in insects and mosquitoes specifically. And that is that, again, mosquitoes are pollinators. They visit lots of different flowers. And if you look at the chemical profiles in mass spec across different floral types, you see that there are lots of different compounds that show up as these little peaks. And you can just see by looking at the patterns, they're different. Now, some of the compounds may be shared in common, but they're different in the concentrations across the entire blend. This floral bouquet, if you will. Now, we can detect that as well, right? So a rose smells differently to you and me than a carnation or an orchid or some other flower, right? We can detect that as well, but oftentimes there are common chemical compounds that are found in floral odors, for example. They're just in different ratios. Well, that apparently is important to a mosquito as well. What's shown on the right here are some behavioral responses.
This stands for performance index, and upward deflections in this graph mean an attraction behavior. So if you put mosquitoes in these kind of wind tunnels and you challenge them with a couple of odors, you find, wow, for some flowers they're highly attracted. I say highly kind of in a relative sense here compared to no odor. And then for some odors, they actually move away. They would choose the other side, no odor. So that seems to have like a repulsive, or we might call that an aversive response in the mosquito. And that's all based on the odor blends that are found in those natural odor sources. Boy, wouldn't we like to know what those are? Now, can you see some of the potential practical applications of that knowledge? One thing that we could do, maybe, is develop better trap systems for surveillance. Right, if we understood the chemical compounds that are attracting mosquitoes and importantly, their blends and the ratios of those compounds together, we could, in theory, I hate to use that term, say anyway, in theory, we could develop blends that are highly attracted to one species of mosquito, like Aedes aegypti. Already we know from trapping systems that are used for surveillance that are used to make decisions about vector control that there are biases. Put a particular kind of trap out, you'll catch one or two or a few different species of mosquito, but you may miss entirely something else that you want to see, that you want to collect, and you want to understand what's happening in a local area. So we think that's one aspect of this information that's important, but what's missing here? Genes and receptors. We do not know, basically, holistically, what any of these compounds activate in terms of their receptors. And so you might say, well, do we really need to know? We could just develop blends that attract mosquitoes without any prior knowledge of what those receptors might be. And you'd be right in one sense, but in another sense, we could also, by identifying those receptors that are most strongly activating the behavioral responses, we could begin to utilize even other compounds that aren't maybe the natural floral compounds that can do the same thing. In other words, mimic those compounds in blends in ways that are more affordable, for example, to use in traps, or ways that are just more practical in terms of their development. So this is why we're trying to understand, trying to give you the rationale of why is it that we care about chemical detection on this particular level. Okay, so here's Robert Huff. He was a PhD student in my lab. He graduated last spring. He's now at UC Davis. And he did a lot of studies on mosquito chemical receptors using the oocyte system. In this case, here's a mosquito, Anopheles gambia, which is one of the primary vectors for malaria in sub-Saharan Africa. And after screening, we have this hypothesis that there was a receptor, it's called OR29, that it would be tuned to linalool. I won't tell you the history of that, but we thought this might be the case. And in fact, it was. And in our compound library of 200 or so compounds, linalool compounds, or closely related compounds, we can argue about how closely related those are or not, but some similar compounds, it turned out that linalool itself, the R plus version of that, there are two stereoisomers of linalool, an R and an S, and the mix of R plus S activated the receptor. These bars are a little bit challenging to see, but they're there. And a closely related compound, tetrahydrolinalool, activated, but essentially no other compounds in our library. So this speaks to what I think is the selectivity or the specificity of the receptor and chemical compound interaction. This may mean for Anopheles gambia that in terms of floral odor detection, linalool, which is a very strong odor, if you've ever smelled it, it smells like lavender for one of the isomers and coriander for the other. So we can actually detect the isomers in our noses, which is an amazing principle too. And the receptors are totally unrelated to one another. Fascinating kind of convergent evolution story there. I'd like to explore that at some point in my career, but not just yet. At any rate, so this is showing that, yes, in fact, there is a selectivity here, even potentially for one or the other isomer of this compound. We've followed up that story looking at yet another receptor. I'll get to that in a moment, but another hallmark of these receptors is that they show a concentration dependency. That is that they're not only sensitive at very low levels of the compound when challenged, but that sensitivity should increase as the concentration of the compound is increased and will reach a maximum response. That maximum response that we see is just simply a function of the total number of receptors that are on the outside of this oocyte. In other words, you could activate potentially all of them, and that would be the maximum response that you could even record from an oocyte. Does that make sense? So we reach that level, and then we can determine this sort of pharmacological parameter called the EC50, or effective concentration at half maximal response. And that gives us a way to kind of standardize across chemicals and receptors. In other words, for sensitivity, which of these compounds elicits the most sensitive response. In this case, when we challenged this receptor with just the R version, this isomer of linalool, we saw an EC50 of about 8 micromolar. I'm just going to tell you, that's pretty good for this system. And then the racemic mixture, which has both isomers in there, gave an EC50 of just slightly under that. Now, at the time of these experiments, we didn't have purified S plus linalool. It's in this mixture. But our conjecture was that, well, if we had it, this receptor would be even more sensitive to that. In other words, the EC50 would be lower. Well, we've actually gone on to demonstrate that, and we've gone on to demonstrate that another related receptor, OR53, shows a better sensitivity to the other isomer. This is a busy figure. What you see here is an alignment of the amino acid sequences, so this conceptual protein, just at the primary sequence level. Between these two receptors, OR29 and 53, these are gene structures which are well conserved, so we think that they're evolutionarily related, and this is a little bit bigger picture of the genome showing that genes surrounding these photoreceptors are also well conserved. So this is just a picture saying that these are orthologous or homologous genes that were derived from an original ancestor gene that was probably sensitive to linalool. Here's the data that shows that OR53 and 29 are different in their sensitivity. So you can see here for Anopheles gambia OR29, S plus linalool gives, in this case, a 7.1 micromolar EC50 and R of 14, so there's about a 2-fold difference. You can see that as a shift in this curve when we compare the CRCs. So linalool here has a shift leftward, and that indicates a lower EC50 or a more sensitive response. Okay, but for OR53, the curve is shifted in the other direction. That is that they're a little bit better, more sensitive, to the R version of linalool. Now that was much more pronounced in this other mosquito, Anopheles stevens. It's a vector for malaria in the Indian subcontinent and other parts of the world. That 2-fold difference in EC50 was evident again, but not so much for Anopheles gambia. We're not sure what this means exactly, but our conjecture is that for Anophelines broadly, these two receptors may convey, OR29 and OR53, differential sensitivity to the stereoisomers of linalool, and that could, in fact, alter behavioral responses to floral odors. We don't have any direct evidence in the mosquito to support that, but that's something we'd like to test. And how might we do that? We might do that by knocking out one or both of these receptors using transgenic technologies, CRISPR-Cas9, if you know something about that, and then test them behaviorally. So that's on our agenda to do, not only for this receptor, but really for any of these receptors. By extension, we are doing this work to try to understand the receptive capacity of these odor receptors in Aedes aegypti now largely. That's the funded work that we have, this collaborative grant. And we're looking at, this is sort of a family tree, if you will. It's a phylogenetic tree based on amino acid sequences. And we've been doing the same kind of characterization for a lot of different receptors. And we find, again, not surprisingly, that each of them responds very specifically to a particular compound, and that the EC50s are in the range that we would expect for a natural kind of ligand-receptor pairing. And so this work, we have on our agenda about 30 odor receptors from Aedes aegypti to do, and then if you expand that to the other colors here, which are other mosquito species that are represented in the hundreds. And the odor receptor gene family is quite extensive, actually, in Aedes aegypti. I didn't say this before. There are about 130 of these ORs. So I think we'll be keeping graduate students busy for a long time doing this work. Just a brief foray into another family of receptors called the IRs. It stands for ionotropic receptor. We think, and we have really good evidence now to support this, not only from my lab, but from other labs that study similar mechanisms, that the IRs are responding to carboxylic acids. Carboxylic acids are fairly ubiquitous in nature. Short-chain carboxylic acids are really important components of human sweats. They're also given off by other animals and tend to signal attractiveness of people to mosquitoes. So boy, we would really like to know what are the receptors for carboxylic acids, and we think that a lot of that lies within this IR gene family. This is just some CRC data showing that, in fact, for IRs from Aedes albopictus and Aedes aegypti, yes, we do find them to be very sensitive to one carboxylic acid very specifically. And in fact, what we do in our library is we have not only nonanoic acid, which is the 9-carbon version of this, but we have all the acids going from like 6 to 14. And it's really interesting, I should have shown the pattern here, but when you start challenging the receptor with the 6-carbon hexanoic, then heptanoic, octanoic, the peak just, it's really sharp for nonanoic acid, peak of sensitivity, and it's almost non-existent for the other ones. Again, that sort of feeds into my own bias of these receptors are really specifically tuned to one kind of compound. So we think this is going to help us formulate an understanding of the basis of chemical detection of hosts for blood meal seeking in females. We also found some odorant receptors in Aedes aegypti that are responsive to carboxylic acid. This was something of a surprise to us. This is, again, Robert Huff's work. And interestingly, Robert challenged those receptors with DEET. And so these are concentration response curves where we increase the concentration of DEET while holding the carboxylic concentration the same across the oocyte. And we see that DEET interferes with the activation, in both cases here, of these receptors, which may speak to the mechanism of repellency. There's lots of work that's been done on DEET. It's an on-the-shelf commercial repellent. You can find it as an ingredient in most of your mosquito repellents that are not necessarily natural mosquito repellents out there. And so we'd like to understand that better as well, because in principle we could use this kind of information to develop novel kinds of repellents as a personal protection measure to reduce biting. Not just nuisance biting, but biting that might actually be from mosquitoes that can transmit diseases. This is just an alignment of some IRs from various mosquito species showing that in this particular case, the nonanoic acid receptor is very highly conserved across mosquitoes. And for these mosquitoes, they're host-seeking humans, principally, so we think this could be a part of that story. Hopefully lots more information to come on that in the years ahead. So some future directions for these projects. We want to continue this functional screening. We have an amazing setup, and we're well equipped to do this. Very few labs do this same kind of functional screening at the molecular level. We want to, of course, look at gene knockouts and do behavioral tests to see are these receptors actually affecting the behavior of mosquitoes in the ways that we think that they are. And then, yeah, test those in olfactometers or various kinds of behavioral assays that we have set up in the laboratory. So I'm going to change gears just a little bit now and talk about, well, what are some of the potential practical applications of this work that we're doing. One of the areas that we think we can make the best contribution to is this idea of attractive toxic sugar baits. Now, I almost hesitated to even put something like this up in front of a group of engineers because it's just a schematic diagram of what an ATSB might look like. And some people have modeled this before. This isn't unique in terms of its design, but the basic premise is this. If you can attract a mosquito, a species of mosquito, even very specifically, to a bait station that holds some kind of reward for them, in this case just sugar water, you can lace that sugar with something that might kill them. This is a model that's actually been used and successfully in some trials to reduce mosquito populations in areas where there's endemic disease transmission. And so it has some advantages in that it can be potentially used to attract, again, through the odor source, only one kind of target insect, and while also leaving the beneficial insect alone, so to speak. So it has this idea of, I'll use the term environmentally friendly. So we're attacking this from kind of two angles. Again, we didn't invent this concept of ATSBs. It's already been out there, but we think we can contribute to better design of them. One is that we'd like to develop odor blends that are more specific through our knowledge of receptor-ligand interactions and what kinds of blends attract Aedes aegypti, and in this case Aedes albopictus, which is also a vector for dengue and chikungunya, and here now in the Americas, Zika virus. And then we want to also go after the toxic part of this, so the T in ATSB, by utilizing unconventional insecticides. In fact, things that are not known to be insecticides at all, that are, this acronym here, GRAS, stands for generally recognized as safe. And that's an FDA designation, and compounds in that category can be used in human products. In fact, one of the compounds I'll describe for you now is found in most products in your home, in your dorm, in your apartment, wherever you happen to live. And that's a really interesting thing to think about, that they have toxicity to insects. So here's just some data from a publication. It's Gunther Mueller and John Beyer have really spearheaded this work over the last couple of decades, showing population reductions in areas where they treated with ATSBs. In their case, they're actually spraying vegetation with a sugar solution that has laced within it boric acid. So that's kind of a common insecticide. And what they find is that even when that spray is dried on vegetation, mosquitoes will still come and feed on it. They salivate after all, so they can dissolve the sugar and then imbibe it, and it's toxic to them. And they show that compared to control sites that are shown in black here versus treatment sites, they show major reductions over time in the population of mosquitoes. So I think this does have some promise in terms of reducing vector populations. What does it do for disease transmission cycles? That's still, I think, yet to be determined, but this is a really interesting model, one way to go about it. So some work in my lab. This is the assay that we use. It's been conducted by Heidi Lindsley. She is now a fourth-year PhD student in my lab. And Melissa Narenya, who was an undergraduate in my lab, who's now graduated and has entered the PhD program in biology at the University of Washington, actually in Jeff Riffle's lab, one of our collaborators. So I'm really happy that Melissa is now going to continue to study mosquito biology. She's the first from my lab at the undergraduate level to do that, so that's super cool. Here's the assay. We take some adult mosquitoes that are about 4 days old. We put about 50 of them in one of these small cube cages, and we give them sugar water in a bottle as a wick. Over time, what you'll see, how long do you think a mosquito can live, say in the laboratory versus nature? Have you ever thought about that? People ask me that question. In the laboratory, we can keep them alive easily for a couple of weeks, a few weeks. In nature, right, so there are lots of things that want to kill mosquitoes in nature. Lots of reasons why they may die otherwise, because they can't acquire resources that they need, for example. So it's certainly at the population level, a shorter lifespan in nature, but mosquitoes can live upwards of a month. And that becomes important in terms of disease transmission cycle. Let me cycle back to that concept of extrinsic incubation period. One of the things that we're attempting to do here is not kill a mosquito immediately. It's not present them with an acutely toxic compound that causes them to die within the first, say, 15 minutes or an hour after imbibing. Why are we avoiding that? We think that by coming up with a strategy that reduces the lifespan of a population of mosquitoes, we can affect the age gradient in a population. That is, we can reduce the number of females that actually live long enough to become infective. They may still take blood meals, they still reproduce, and we think that gets around a lot of the natural selection pressure that leads to insecticide resistance. So the concept here is not to kill a mosquito on first sugar meal, but to just shorten their lifespan on average. Okay? So here's what we do. Over a period of 7 days, we monitor viability. We just count the dead ones. I said, count the mosquitoes each day, but really we're just counting the dead ones. How many are alive after 1 day or 2 days or 3 days? After we present them with a sugar solution, and this is important, there is sucrose in this bottle, but we lace it with some other chemical compound. I had done some previous studies where I found this compound, propylene glycol. Have you heard of propylene glycol? Pick up your shampoo or your soap or your toothpaste at home and look at the ingredient list. Propylene glycol will be there. Yeah? So it's reasonably safe, apparently, for human consumption. But it turns out that even at low concentrations, and here's a range from 5 to 10% that we've used, when you lace sucrose with that, it kills mosquitoes. It's toxic to them. We don't know why. Mechanistically, we have no idea. That's okay, we don't need to know why just yet. We'd like to figure that out eventually. So you can see just kind of these survivorship curves over 7 days. They drop off. This is a control with sucrose only, as I described to you. Most of the mosquitoes are still alive at the end of that period of time. We've modeled this in 2 ways. One, where we provide this compound in sucrose continuously over the 7 days. That's the ad libitum side of this. But we've also modeled this with just a single feeding. We present this compound in sucrose for only 24 hours, then we replace that bottle with sucrose only. We did that because we thought, that's going to mimic maybe a more natural scenario, where a mosquito may only visit your bait station 1 time, right? And then may utilize other nectar sources for sugar meals after that. And maybe most of the population won't visit your bait station at all. We hope the odor side of this lends itself to increasing that uptake of the sugar solution. But this is the idea, and what you can see in the graph on the right here is that, In fact, with just presenting that sucrose for 24 hours, still over time, the average lifespan was reduced in these laboratory populations in mosquitoes. So we think that's an important component of this in terms of modeling how this may work in a more natural setting. We've done this for a few different mosquito species, Aedes aegypti, Aedes albopictus, and Culex pipiens. That's the West Nile vector. And we actually found, a little bit to our surprise, differential effects. Culex was way more sensitive to this compound, this dialpropylene glycol, than the other two species. But actually, Aedes albopictus was quite sensitive too. It's remarkable, even after just the 24 hours of feeding. So, that says to us that maybe not all of these toxins are, of course, going to have the same effect on just general populations of mosquitoes, let alone even potential beneficial insects. We've not yet tested this on things like honeybees or moths or butterflies. We hope there are some engineering controls in these ATSBs with screening that wouldn't allow larger organisms to even feed on the sucrose solutions, but that's something that yet we'd like to understand. We've also moved on to test other diol compounds, so these compounds that have two OH functional groups attached to them, and we find similar results, that for many of these compounds it reduces lifespan. This is in an ad libitum scenario. And then we've also found that artificial sweeteners, aspartame, butane, erythritol, xylitol, ACE-K, stevia, some of the things that you also might find in your non-sugar sweeteners out there that are generally recognized as safe, have toxic effects in mosquitoes. The advantage for us here is that we think we could use these compounds pretty much right away. Right? They're already recognized as safe for human consumption. If we can demonstrate that they have maybe reduced effects on beneficial insects, then the regulatory process for developing a new toxic compound for insects could just really stifle this work for years and years. But by using this kind of approach, we think we might find things that are much more amenable to use in the immediate sense. Okay, so future directions with this would be now to really work on those odor blends to try to attract Aedes mosquitoes specifically, and then continue to screen, especially the generally recognized as safe compounds, and test them at different concentrations so that we come up with a formulation that we think is applicable very soon. I just want to very briefly, for a minute or two, I think I'm almost out of time here, just mention another project that we're initiating now in houseflies, Musca domestica. Houseflies, of course, are commensal with humans. They're found literally every place that humans are found. Did you know that at the South Pole Research Station, they have houseflies in the kitchen? That kind of really surprised me when I heard that. Like, okay, so houseflies are everywhere, right? They are. We know them as nuisance flies, but houseflies are also potentially very important, what we call mechanical vectors, transmitting bacteria especially, but other kinds of pathogens, just because they like to live around filth. They land on garbage, on trash, on decaying organic matter, on fecal matter, right, defecation. So they land in all these places and they pick up these microorganisms and they carry them either on their mouth parts or they pass through the gut system and they defecate them. I know, this is kind of gross to talk about, sorry. But they also carry them on their feet, on their tarsi, right, to human food. You've been at a picnic, you've had houseflies landing on your food, right? And people ingest them, and many of these pathogens are entopathogenic to people, so they cause dysentery. So we want to study olfaction. It's arguably understudied in the housefly. We're doing the same kinds of experiments where we want to know for these odor receptors in this large family, what are the compounds that activate them? I'm just going to say, in summary, we found that indolic compounds activate some of these receptors that we knew already from mosquitoes. But the last important thing I want to say is about John Boye's work. So this is kind of more of the same information. CRCs, responses in oocytes, highly specific responses to indolic compounds for these receptors. But my second-year graduate student, John Boye, and I have become very interested in pheromonal detection. We found in assaying expression in the antenna, we can do this globally, so looking at all transcripts that are there, that there are some transcripts that are differentially expressed between males and females, highly differentially expressed, which already suggests that these receptors are somehow involved in mating behavior. Okay, so why would we say that? Well, this little group of receptors are those receptors that are differentially expressed. They are very closely related to a receptor from fruit flies, Drosophila melanogaster, that's already a known pheromone receptor. Cis-vaccinyl acetate, it's this large hydrocarbon. Well, there's a hydrocarbon that's known in houseflies as a pheromone that is attractive to males and is an instigator of mating behavior. So we think these receptors may be sensitive to muscular or the C9 trichosine, and that's a project that we're going after very hard now, because if you can interfere with mating behaviors in organisms like flies, that can really have a very rapid population depressing effect. So we'd like to understand first and foremost the biology of this at the level of chemical detection, and then hopefully be able to utilize that information for developing new ways to control houseflies, not only for their human impacts, but even for their veterinary impacts as well. So I'm going to stop there and just say thanks again to all the people in my lab and our collaborators who have really been just wonderful to work with through all this work. Thank you for your time. I'm happy to take questions if you have any.
So is it correct to say that there's protein gates and receptors?
Yes, they are. Yeah.
And so odors bond to protein
Mhm.
gates and cause them to open.
Yes.
How are those protein gates receptors?
Yeah, that's a great question. Okay, so the question is, are these protein gates, these channels, these ligand-gated ion channels, and how are they reset? Great question. So there are a couple of mechanisms for resetting. Yes, so these chemical compounds bind to the external surfaces of the proteins, cause them to open. There are enzymes that are found in that syncytium lymph, that liquid-filled cavity in the hair. They're actually odor-degrading enzymes, and they act on those compounds to break them apart, and once they're broken, then they don't actually activate the receptors anymore. And there are also mechanisms to transport them out of the syncytium lymph as well. So there are a few different mechanisms. Truly, they're not very well studied, I would argue, but they are there for relieving this activation, because that would lead to a lot of noise too. Imagine, right, if the ligand activated this receptor and just kept doing that, there would be constant action potential signaling to the brain, when maybe the compound is no longer found in a particular environment. That resetting happens at very, very rapid scales, milliseconds.
Oh, so it's very fast.
Yeah, the latency is really fast in terms of the response and the shutting off of the response in neurons. Yeah, and these receptor neurons. Good question. Yes, please.
I know it may seem very unscientific, but it seems like some people attract more mosquitoes than others. Like my mother goes outside and
Yeah.
she has a swarm around her almost
Yeah.
constantly. Have you looked into maybe some people produce different mosquito-attracting chemicals? Have you looked into that?
It's a great question, actually, right? And it's one that people who work in my field get a lot, as you imagine, because everybody has this anecdotal experience. Like, why is this person much more attractive to mosquitoes or other biting insects than others? Anecdotally, we recognize that to be the case. There's really good evidence, experimental evidence, that supports that hypothesis. We think, of course, that it has to do with odor profiles. We smell different, right? My sweat doesn't smell like yours, exactly, especially to a biting insect. But we really don't have a handle on what are the specific compounds or the ratios of those compounds in something like human sweat that determine that behavioral difference that is quite evident. I always jokingly say it's like the old adage about I don't have to outrun the bear, I just have to outrun you. So take your mom with you when you go on hikes or camping, and you'll be fine, because all the mosquitoes will go to her. Right? It's a great question. It is something we are certainly interested in, but we'd like to start to understand the molecular details first, and I think we can work our way up to that.
So
Yes.
if you get a mosquito repellent, like your standard DEET spray that you
Yeah.
put on yourself, what is the way in which that modifies the story you're telling up here and
Mhm.
keep them away?
Yeah, so this steps into, if you're in my field and you go to a scientific meeting and someone asks that question,
Yeah.
it becomes very controversial.
Okay.
Let me just say this in summary. There are multiple modes of action for DEET.
Okay.
It can repel mosquitoes at a distance, we think largely to interfering with the ability of certain compounds to activate receptors, but it's also a contact repellent. There's some neat experiments that have been done in mosquitoes where they knock out these odor receptors globally. But the taste receptors are still intact. The mosquitoes now will not be repelled at distance. They will still land on a human hand, for example, but once they taste the compound, they fly away, they don't bite. So that clearly says there are multiple modes of action for DEETs, even more than I'm not describing here potentially, because they repel ticks as well, and ticks do not have odor receptors. So they're still repelled at distance, and they're certainly repelled by taste, but we don't understand the basis for that. There's more work to be done there. It is a great question. Yeah. Yes, sir.
So you
mentioned toxins that target the organisms of old.
Mhm.
Do you think it is possible and feasible to synthesize a toxin or chemical that would target either neurotransmitters of the neuron or even the protein channels, locking them open or closed so there would either be no input or too much?
Yeah.
Is it possible to do that? Kind of like Dieter.
Yeah, so is it possible really to alter the mechanics of like
Yeah.
these ligand-gated ion channels to affect repellency?
Yes, sir.
And that is it's a super question because it's one of the major sort of hypotheses in our field. It's called excitorepellency. Can you hyperactivate a receptor that stays on all the time? Maybe these odor-degrading enzymes aren't acting on that particular compound, and so the receptors stay open too long. That creates confusion in the organism, so it's no longer able to hone in on its host, whatever that may be. So yeah, there's good reason to believe that that is going to be the case for the development of novel kinds of repellents. Yeah, and there's some really great publications on that concept specifically. Yeah. Does that mean it's over? Is that the
No, we got time for one more question.
Another
One
question.
more, one
Okay.
more. All right, well, let's thank you for coming. If you'd like to join us for lunch, we'll be at the Corner Cafe in the Joyce Room in 10 minutes.